So welcome everyone, and thank you for virtually joining us for the latest version of the Variant Effect seminar series. My name is Yanil Boudot. I'm a member of the VES organizing committee, along with Adrini de Souza and Diego Calderon. These seminars are made possible with the support of a lot of help from Lara Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas of Variant Effect Alliance. As a reminder, we're always on the lookout for potential speakers for future seminars. If there is anyone you would like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentations, I have a few housekeeping items to mention. Each talk is around 20 to 25 minutes, but there should be time for a few questions. Please post those questions at any time to the Zoom Q&A, and we'll read them out to the speakers at the end. If you have additional questions after the end of the seminar, feel free to join our seminar series Slack channel or discuss with some of the AVE members. And finally, we will be posting this seminar to our Twitter account. You can catch the seminar to our Twitter account through our YouTube account using the CMAP channel. And don't worry, we're trying to copy the URLs. They will be posted in the chat once we get started. All the details of the seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. For the last housekeeping item, there is rarely an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. So up next, we'll have two excellent speakers. The first is Willow Coyote Mayetes. He's an assistant professor in the bioengineering and therapeutic science at the University of California, San Francisco. His lab focuses on building a biophysical, molecular, and cellular understanding of the secret of the life of the membrane proteins. They build and apply scalable technologies to learn how membrane proteins underlie our physiology, what goes awry in disease, and how to better treat this disease with therapeutics. Willow, whenever you're ready.
All right, thanks so much to the organizers for this opportunity to share to this community. And in the introduction, I'll be talking a lot about the work we've been doing the last couple of years to use mechanistic genetic screening to really start uncovering the basic biology and translational biology of membrane proteins. And so I've become fascinated with membrane proteins over the past many years due to their role as serving as a molecular and interface between the chemical and physical environment outside the cell and the inside of the cell. And that allows membrane proteins to really play fundamental roles to underlie our complex sensory physiology so we can perceive of the world, so we can maybe understand each other's sciences, but also so that we can live at all. They underlie how we can think, but also like our heartbeats and the rest of our electrically excitable tissues. And of course, the membrane proteins sit at the interface between the inside and the outside of the cell, which means they interact with the vast majority of pharmacologies, either directly or indirectly, that we use to try and treat our disease. And unfortunately, mutations within these membrane proteins have really underlied the immense diversification of our evolution, but really, unfortunately, they've also they cause membrane proteins to break in disease, and then they also disrupt the efficacy of drugs that we use to treat these diseases. And there are many different mechanisms by which a given mutation can have its impact. And as a community, the variant effects community, really thinks a lot about the effects of mutations, but I also want to urge everyone to think about the mechanism by which a mutation has an effect, because it spans scales from the biophysics of folding to the cell biology to trafficking of all the way structure-function relationships. And if we want to understand and predict how mutations have effect, we really need to understand how those effects are happening. But I would argue that a real problem here, especially in membrane protein space, is that typically the approaches we have really lack throughput. Mostly membrane protein biologists use really hard biophysical approaches or cell biology approaches. or they're insufficiently mechanistic, which really means that we don't learn how mutations or other genetic variations have their effects. And so my solution is to really push forward mechanistic genetic screening of membrane proteins. And what I mean by that is, if you think about a genetic screen, most genetic screens really sit at the top here. And many of you do beautiful work, and I really admire it. But sometimes if we're just measuring a single phenotype at a time, cell growth or another phenotype, we're really losing out on a lot of the information that's encoded in this data and encoded in our protein systems. And that's because for a membrane protein, for example, to play its role in cell physiology and cell fitness, it first needs to translate, it needs to fold, and yada, 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 till it can play that role. And by mechanistic genetic screening, what I mean is by measuring multiple different phenotypes across the stack, and then we can learn how mutations or how a genetic knockdown breaks a system. And so we're using these sorts of multi-phenotypic readouts to really learn how perturbations change membrane proteins mechanistically, so we can combine mutations and gene knockdowns in the context of different small molecules and other stimuli to learn how proteins function, how they fold, and how they traffic, and then how they break in disease in these contexts. And then using that to really understand how they underlie our physiology, how they break in disease, and how we can better treat these diseases with pharmacology. And so now I'm going to transition to one of the events I'll talk about today that I'll probably spend the most time on. And this is a story that started about 2 years ago, working in collaboration with Kathy Giacomini's lab at UCSF. And Kathy's really been a pioneer in cloning transporters starting in the 90s and maybe even in the 80s to find what they were doing in physiology, and then translating a lot of that work in guiding the FDA and approving drugs and looking out for drug-drug interactions or pharmacogenomic interactions. And so transporters like this one, which is a cation transport, move many of the drugs we know, like morphine or metformin, across the membrane. However, polymorphism these transporters, like one, change whether the transporter can function or not. So if it's a function, they get into the cell, or maybe too much will. And that can lead to negative side effects. Too much drug accumulates in tissues, or the alternative, where a patient's no longer being treated. There's a VUS problem that many of us will be familiar with, but there's also a basic biology problem where we don't understand how these transporters work. And I would argue that the mutational scanning that we all do can really solve both of these problems. So what we did is we developed a functional assay, two different functional assays. One many of us will be familiar with, which is VAMP-seq, which is an expression-based readout, so we can differentiate mutations that affect expression, and then one that uses a cytotoxic substrate, so we can identify substrates that break mutations that break function. And then this work was really pushed forward by Sukwa, a fantastic scientist in Kathy's lab, who's now a professor at UCSF, and then 2 graduate students and a postdoc in my group. And what we did is we used our approach DIMPLE to make a library of OCT1, and then we made stable cell lines with that library, and we measured how each mutation affected expression and function. And then we could use that data to then stratify the variants into these different classes. And so now what we're looking at is 11,000 points, where every single dot, of course, measures is a representation of a mutation that will either affect expression or function or neither. And so you can see there's this inverse relationship between the two, where if something doesn't express, it's over here, and then it's not functional as well. And then we can draw cutoffs using these distributions. And then what we find is using those cutoffs, we can identify residues that break based on a specific phenotype. And we can identify residues that are important then for that phenotype. So for instance, all these residues that are high are involved in folding instability because they have low abundance in VAMP-seq. And we can map this onto the structure. And at this time, we had no solved experimental structure of this protein. And if you don't know, membrane protein folding is a very hard problem. And historically, we haven't really known how to map how proteins break folding in membrane proteins. But with mutational scanning, we can unambiguously map this, and we find that there's this contiguous backbone of stabilizing residues that connect from the inside to the outside and really cover the first 6 of 12 transmembranes here. And in fact, we identify an alpha helix that's very sensitive to mutation that encode for various stabilizing residues. There's negative charge residues that make disulfide or that make ionic interactions. There's a cysteine that makes a disulfide bond, and there's aromatic residues that pack in here and make interactions with the tops of transmembranes. And you can find it's very conserved. So we've identified a new kind of hallmark motif of SLC22s, or this whole subfamily that doesn't involve just OCT1, but about 20 different transporters. We also wanted to identify the functional determinants, so we also drew cutoffs, and so those are in blue over here. However, without multiple structural states, it's really hard to learn how residues change and impact the conformational cycle. So with this, we turned to some collaborators in Sweden who have this incredible, who are pioneering a lot of the methods in predictive structural biology for membrane proteins. And I won't go into the details of it necessarily, but effectively you can use coevolution matrices to identify residues that are making contacts in some states and not others, and then apply machine learning to this, and then relax everything down with molecular dynamic simulations. And then from that, we get this whole landscape where every single one of these blue areas is a stable state. And then we can take that state and ask, what is the structure overall at that point? And we can go through and get the full conformational landscape without a substrate. And then we can repeat that exercise with a substrate, MPP+, which is very similar to our substrate in our screen, and we can get the entire conformational cycle. So this is starting from just an AlphaFold structure and then enumerating all the different conformational states based on the physics using machine learning and molecular dynamic simulations. This is really pioneering stuff, and it means we can start to do structural biology without needing experimental structures, maybe. So then next what we did is we developed a score for residues that are important for conformation specificity versus function specificity. And we can compare the two and find that they're very, very similar qualitatively, as you see up here, but also quantitatively. And what we find is that these residues, we started to want to stratify the residues and identify what they're important for. And we find that they're actually overwhelmingly mostly important in substrate binding. But then what we also wanted to do is see if we can identify something new. And we found that there's these two transmembrane domains, TM2 and TM11, that make interactions in one state and not another, as can be seen down here in the inward state. They're making a lot of contacts, versus over here they're not. They're also enriched for functionally important scores. And we can see that when you look at residues that are making interactions over here on the right, you find that they're very sensitive to mutations overall. So we're able to discover a new motif that's important in function in this. But as we're going this, if you know much about the structural biology field, you know it tends to be a very competitive space. Many other experimental groups, I guess, were solving structures as we were doing this completely independently that we were very surprised when they came out. But it was kind of fun because we first got one structure, OCT3. Then we got a series of other structures that came out in June of this year. And then we got our manuscript out at about exactly the same time as theirs. And then one other group came out. And so what this allowed us to do is we had all these experimental structures that were independently generated from our computational structures. So what we can do is we could just compare them across each other. And what we find, to our incredible surprise, is that they are so similar to each other. So in blue here is the RMSD, or similarity between structures. And we find that the solved inward states that recently came out like 4 weeks ago are really similar to our computational structures that are inward state. And in contrast, the outward states that were previously solved are very similar to our outward states. So that means we can accurately predict structures, and you can see overlapping one over here on the right. But we also had one substrate, or someone solved a structure with the same substrate bound. And so we could actually compare how well we did at placing the substrate in here. So in blue are our residues, in yellow are their residues, and these are the two different substrates. In green is ours, and in orange is theirs. In other words, we can accurately place a substrate in a computational model without any experimental data at all. And then, of course, with the DMS, we can start to bridge the structure to the function. So finally wanted to bring this in and ask the question, do we see results here with human physiology? Because we had characterized virtually all the VOSs. And so what we did is we went, Sukhwa went down to John Whitty's lab at Stanford, and she learned how to use the UK Biobank data set, of course, which is hundreds of thousands of genomes with patient health records. And we know OCT1 function is associated with LDL levels. So we could simply just ask the question, in our screen, are variants that are low functioning, also low LDL levels? And we see indeed that's the case. And so I've shown you how with a simple multi-parametric assay, we've been able to identify the residues that drive folding in a transporter for which we did not know how it folded. So we've uncovered really basic biophysics. We've also been able to do structure function work without experimental structures and really validate that extensively. And then finally, we've shown that all of these insights that we learn about the mechanism translate to people, and we can start to predict things such as if you know a mutation is breaking folding, it's going to affect all drugs likely. Whereas if you know it's going to affect function, it might affect certain drug response versus others. And so I hope I've convinced you of the utility of mechanistic work in this space for doing really beautiful basic biology, but also potentially enabling clinical genetics. I'm going to switch gears now to another story I'll tell you very briefly that's about GPCRs. So GPCRs are fundamental sensors, they're membrane proteins as well, that allow us to sense the world around us. And they really are the primary sensors of ourselves. And so for a GPCR to function, it needs to first be expressed, of course. It needs to make it to the surface through trafficking. And then after activation, it tends to be internalized. It'll be degraded. That's a desensitization mechanism. But then you'll also get a downstream signaling. And of course, mutations can affect each of these steps. And so that can create disease, but also affect the efficacy of drugs. And so we started this project, and by we, I mean an amazing student in my lab, Matt, about 1.5 years ago. And when we started, we didn't have really great fluorescent readouts for GPCR activation. And this had been a real challenge for the field overall. And so this was a transcriptional assay that we started with, where you activate the receptor, you look at downstream signaling. And when we started in Matt's rotation, we started, and this is a DMSO vehicle, then you look at a superagonist up here, and they kind of all look the same. But we could see a slight difference with an inverse agonist. We turned to a bioengineer and protein engineer, Justin English, at University of Utah, and his great postdoc, who developed a library of transcriptional response elements, and then we added some other engineering handles that allowed us to optimize this to the point where we can see different types of agonists. So we can do real pharmacology now and be really confident in the responses. And then we've done this in other receptors. So this is beta-2AR, which is a classic model receptor. We worked on a proton sensing receptor, which gets activated at different pHs. And then finally, we showed we could do this with an mu-opioid receptor, which is actually an inactivating receptor. And so what this means is we can broadly apply this. We've done this in many other systems thus far, and we find this is broadly useful and very sensitive assay. So for the first application, what we turn to are these proton sensors. And so Ashish, who you may not know, is an incredible GPCR structural biologist at UCSF. And he had this great grad student, Nick, who is trying to understand how proton sensors sense, working with the XP Wang, who's at UNC Chapel Hill. So you have all these proton sensors that allow us to sense the acidity of, or our tissues to sense the acidity of our blood and other parts of our physiology. And so you have a set of different GPCRs that are tuned to different pHs. You have ones that activate at basic pHs, which is GPR4, and those that act at like acidic pH, like GPR68. And they did this chimeric study where they were trying to figure out how much, like, is there a specific hotspot that would be important in pH sensing? So they simply took GPR4, which activates again at pH, at basic pH, and they just started swapping parts. And what they found is that you could swap all the extracellular loops and you get most of the way to an lower pH activation, but you actually need the whole GPCR. So for a conventional structural functional biologist, this is very frustrating because they were trying to answer this question, which is, what is the molecular basis of proton activation? They solved all these structures. This is an immense amount of work. If you know about cryo-EM, it takes so much effort. It's really expensive. They solved about 8 or 9 structures at different pHs with different ligands, bind cofactors. And you can look at all the extracellular regions, and they have lots and lots of ionizable residues, which could be involved in pH sensing. So in other words, there's many residues that could be protonated. There's many different models of how they could be activated and many different experimental models that cause many headaches. They did 300 dose response experiments to try and uncover this, and they couldn't figure that out. And so when we came in, we realized what we need is we need an unbiased functional readout of how does every single mutation affect proton activation so we can compare across everything. So we used our cyclic AMP response element system that we could sense the difference between a low pH and a high pH condition with very good sensitivity. I mean, this is, I think, very beautiful sensitivity for an assay like this. And then we can do a dose response curve, or effectively an endpoint assay. We actually did 3 points along this curve. And we can pull this out, and we find that we get this really beautiful heat map, of course. I won't spend a ton of time on this. Many of you are familiar with them. They're very rich data sets. But we run into this problem, of course, which is we want to learn how mutations affect activation, so we need to differentiate the effects on expression versus signaling. So we also did a mutational scan for surface expression using an epitope tag on the outside of the cell. And then we have a readout for surface expression and function, which we then can fit the synonymous distribution as a line, and then we can just look at what's above and below this line. And we did that. And then from that, we get this unambiguous loss of function, gain of function kind of score that's normalized. So we have their structure, and we wanted a structure function map, use the mutational scanning for the function part. So we took the structure, we generated a contact map, which if you're not familiar with, what this shows is in 2D space, what are each of the contacts between residues. And so a black spot represents a contact between, say, position 25 and 175 here. We can map on our loss of function scores, and we can also map on our loss of function scores onto this contact map. And then what we get is a functional contact map, because we can look for where there are places that interactions are being made. And then what this does is it reveals a network where you find here is histidine 269 that comes across, and it makes interactions with this other histidine H20 and this tyrosine here and this E as well, which is making interactions in with this arginine. We can look at this, and this is actually a very close network of residues that appear to be essential for proton sensing, and they're making interactions. There's even more above and below that connect all the way from the top to the inside of the receptor, which you can see you get the top here, and then you get some in the middle, which gives you the signaling response. Then what we did is we picked up specific mutations to make validation mutations, and we find really great sensitivity where we can make different changes to pKa, like making a positive charge will shift the pH direction in one direction versus the other. First, we can break this ionic interaction and really break the activation as well. So I showed you that we can use this approach to identify the distributed network that contribute to the emergent property proton activation in this proton sensing GPCR to really do integrated structural biology. And we've really shown that it's a distributed mechanism and been able to identify some of the key players in that molecularly. So I'll go through this pretty quick because I think I was disconnected, so I'm a little slow, but we had a sensitive transcriptional response assay that works broadly for GPCRs, which allowed us to unpickle a hard cryo-EM project using a DMS for proton activation, which allows us to get to integrated structure function biology maps. We've done this now across a bunch of different genes, including a channel before, the transporter story I showed you, and now a GPCR. So this is broadly applicable for use in structural biology and biophysics, but then also we can use the data for clinical genetics, which many of you will care about, obviously. And many different mechanisms affect the effects or underlie the effects of mutations. And so we're trying to push forward broadly on this effort. And we're doing that by developing new technologies, generating many different data sets. And then we've even started creating new statistical frameworks in collaboration with some friends at UCLA, Harold, and his star student, Jing Yao. And with that, I'll just say thank you. I acknowledge everyone here, acknowledge you for giving me this opportunity and listening to me ramble. I'm hiring a lot, so if you need, if you're looking for postdocs, please reach out. Also, we're hiring a faculty position in my department. So if you're looking for faculty positions and you're at the end of your postdoc, please apply. It says that it's closed. It's not closed. We'll be looking at applications for the next couple of weeks. So Yeah, come be my friend, and happy to take questions.
Thank you, Willow, for your amazing presentation. And I think we have time for a couple of questions. So if you're wanted to have a question, please post here. And we're gonna give some minutes so questions starts to pop up. But in the meantime, I think we have some questions from Diego. Diego is asking what you think there are the different classes of proteins that might be the most promising to target with this kind of approaches.
Well, I'm really obsessed with membrane proteins, so I would say membrane proteins. But I think like, yeah, membrane proteins to me are the important one to target for me. And that includes GPCR transporters and channels, which is what we're doing. I would say, it's always about, as many of you will appreciate, it's about finding like, do you have a good readout, and then are you sensitive enough to detect the difference between how mutations are affecting things, and then is there a good biological story to tell too, that intersects with that.
Great.
Yeah.
And I have a question. I'm wondering if your model could also be used to look at variants that are in cis or like pairs of mutations or triple mutants, if you think that would be also efficient, and if you consider studying epistatic effects.
Yeah, I mean, so it would be useful. I think that there's a lot of technical challenges that needs to be solved to get there, because the vast majority of mutations aren't epistatically interacting, so it's and as many of you will appreciate, and membrane proteins are quite large, and so they're like, you can't read out the multiple positions and you can't make all the mutations. So we have to think a lot about like how we might choose mutations to make, and which residues to choose. And I think there's a lot of information out there with coevolution, structural information that we could use to start to do that. And we're really excited to do that, because I'd say it's one of the biggest challenges for protein engineering, and also for understanding the biology. I'd say less for a lot of the variant stuff, or the variant prediction, because just combinatorial mutations are less prevalent, but it's definitely also important. So yeah,
amazing.
Absolutely.
I don't see any more questions here. Let me see in the panel in the Q&A. Yeah, so I think we can go ahead with our next seminar and our next talker. So thank you so much, Willow.